Meiosis is the process of creating gametes, which are sex cells. Meiosis, however, is not a cycle like mitosis. There are two cell divisions, meiosis I and meiosis II, which result in 4 daughter cells, each containing 23 individual chromosomes. Interphase is a stage between divisions. Basic cell growth, replication and repair of the DNA, and final preparation for cell division occurs at this stage. Phase 1 is when homologous chromosomes from each parent pair up to form homologous pairs. Spindle fibers also attach to the centrioles. Process that occurs during prophase 1 is called crossing over. This happens when parts of the homologous chromosome switch places after overlapping. Crossing over increases genetic diversity. The next step is metaphase 1. This is where the chromosomes are lined up at the metaphase plate and spindle fibers are attached. They are lined up in preparation to separate into 2 cells. There are more than 8 million ways that the chromosomes could orient themselves. Anaphase 1 involves the centrioles using the spindle fibers to separate the homologous pairs to the opposite poles of the cell, but the sister chromatids are still attached. A non-disjunction causes the gametes to have the wrong amount of chromosomes. As seen in this picture, the spindle fiber did not attach correctly to the chromosome. This will lead to an abnormal amount of chromosomes in each cell that is produced. Seen in this model, the chromosome that was not attached to the spindle fiber will now be pulled to the opposite cell, causing 3 chromosomes in one cell and 1 in the other. In the end, this non-disjunction will lead to 4 daughter cells, in which all have the wrong number of chromosomes. 2 of the daughter cells will have 1 chromatid, and the other 2 will have 3. An example of a chromosomal disorder that involves this type of non-disjunction is Down syndrome. Down syndrome is a chromosomal condition associated with intellectual disability. Most cases of Down syndrome involve a third and extra chromosome 21, hence the disorder's other name, trisomy 21. Many researchers believe that the extra chromosome disrupts the course of normal development. People with Down syndrome have abnormal facial features and weak muscle tone at infancy. As they grow, these children have learning difficulties and are slow at meeting developmental milestones. Down syndrome is a fairly common disorder with more than 200,000 U.S. cases per year. Now let's go back to our normal cell. Telophase 1 is now occurring and the cell creates a temporary nucleus around the 2 homologous chromosome sets. Cytokinesis then occurs and divides the cell into 2. After meiosis I, meiosis II occurs in 2 cells at the same time. Meiosis II starts off with prophase II, which involves a chromosome being connected to the spindle fibers. In metaphase II, the spindle fibers line the chromosomes up to the metaphase plate. As you can see, this is much like metaphase I. Then, in anaphase 2, the centrioles use the spindle fibers to pull apart the chromatins to opposite poles of the cell. If the centrioles do not properly attach the spindle fibers to the chromosome before they start to pull, a non-disjunction can occur at this point too. At the telophase stage, the cell creates a permanent nucleus around the groups of chromosomes. Finally, the entire process ends with cytokinesis, when both cells split into two to create 4 haploid cells. And that is the process of meiosis.